So let's get started with Prophase 1. We'll start at the very beginning because after all, that is a very good place to start. So the things that are similar in Prophase 1 that we also saw in mitosis is that we have to get rid of that nuclear envelope. That pesky nuclear envelope is in the way, so we got to get rid of that. Okay, that's the same. The other thing that we're going to see the same between Prophase 1 is that we're going to see our DNA go from a chromatin form, right, that spaghetti form, to condense down into our chromosome form or the Cheeto form. Talk about some really important differences that occur in Prophase 1 that we did not see in mitosis. Okay. During Prophase 1, we see synapsis happening. This is when our homologous chromosomes are going to become in quote close association with each other. They're going to boink, kind of attached to each other, and they're going to be held together with these special proteins that are going to keep them kind of tied up together. Okay, so that's called synapsis, where our homochromos synapse together. And this is important that we have these homologous chromosomes being held together with these special proteins so that we can get to this process called crossing over. Okay, so let's look at synapsis a little bit closer. So during synapsis, the chromosome that we would have got from dad is going to hook up with the chromosome that we would have gotten from mom. They're synapsing together. They're being held together by these special proteins. Remember that the replicated copies are called sister chromatids. Write this big in your notes. Stars, highlights, big arrows, whatever you do to make sure that this is really obvious in your notes. The second source of genetic variation comes from crossing over. This is where we're going to see homologous chromosomes swapping portions of their chromosomes with non-sister chromatids. For our sister chromatids, we're going to swap a portion of this chromosome with our non-sister chromatid, right? If we switch with our sister chromatids, it wouldn't matter because there wouldn't be any genetic variability. But what we're going to take is a portion of that chromosome that came from mom and swap it with a portion of the chromosome that came from dad. Okay, now we have a novel combination of genes that didn't exist before in either parent.